repolarization, just as we had with the action potentials that occur along neurons. Okay, so you have a series of events where you have first opening of voltage-gated sodium channels that cause depolarization, then opening of voltage-gated potassium channels that repolarize that segment and spreads across the sarcolemma. Okay. So, once it's propagated, the action potential travels down the T-tubules. When it reaches the triad, that segment where the T-tubule is surrounded by terminal cisternae, it triggers release of calcium into the sarcoplasm. Calcium is going to bind to troponin, okay, which then displaces the tropomyosin from the binding sites of actin. It uncovers them. That allows cross bridges to form. So myosin cross bridges alternately attach and detach. Okay? And with each cross bridge formation and each power stroke, the thin filament is moved to the center of the thick filament. Okay? Hydrolysis of ATP powers this process. In other words, ATP has to keep binding to the myosin head, causing detachment. And as it's hydrolyzed, that power, that energy is used to then cock the head so that that series of events can be repeated. Once the calcium is removed back into the sarcoplasmic reticulum, again, this is powered by ATP, the muscle begins to relax. Why? Because the binding sites on actin become recovered by tropomyosin. And this slide kind of demonstrates that same sequence that we keep talking about. Here's the action potential traveling down the T-tubule, releasing calcium. Okay? That calcium binds to troponin. Okay? It displaces tropomyosin, cross bridges form. You have that ratcheting action. And we get this line of the filaments until the calcium undergoes reuptake back into the sarcoplasmic reticulum. Okay, and we can define each step in this pathway. Okay, cross bridge formation is simply where the myosin forms a linkage with the actin. We talked about the power stroke. It's when the myosin head pivots or ratchets, pulling the actin filament toward the M line. We said that cross bridge detachment is brought about by the binding of ATP. Okay, and then when the ATPase along the myosin head cleaves that terminal phosphate group off, that provides the energy for cocking of the myosin head. And this whole series of events repeats itself. And this is just some definitions with regard to contraction and shortening and how contraction ends. Okay. Earlier we talked about, without using the term specifically, we talked about motor units. A motor unit is a motor neuron and all the muscle fibers it supplies. Okay? The number of muscle fibers per motor unit can vary from 4 to several hundred. Let's go forward for a second. Okay? What I'm showing you here is simply two different axons being carried by the same nerve to a particular muscle. As each axon approaches the muscle, the organ, it branches, and each of these axon terminals provide innervation to a different muscle fiber. In this case, this motor unit provides innervation to 1, 2, 3 muscle fibers. That's a relatively small motor unit. Likewise, this motor unit, which provides innervation to 1, 2 fibers, is also very small. On the previous slide, we said that most motor units provide innervation to somewhere between 4 and several hundred muscle fibers. Okay? Muscles that are capable of exhibiting fine movements, like the muscles of the fingers and hand, or those of facial expression, are associated with small motor units. In other words, axons provide innervation to relatively few muscle cells, whereas the large weight-bearing muscles, like those of the hips and thighs, like the gluteus max, have huge motor units. such that one axon provides innervation to hundreds of fibers. So of course those large muscles aren't capable of very much precision, only bulk movement, weight bearing, walking. Now, those muscle fibers innervated by a single axon, they tend to be scattered throughout the muscle, not limited to one side or another. Okay? So contraction of a single motor unit causes very weak contraction of the entire muscle. Okay. When I took physiology in college, we did muscle twitch experiments. A muscle twitch is a response of a muscle to a single brief threshold stimulus. It's usually some type of electrical stimulus. And what we did in college is we took a muscle from what would be the calf of a frog, okay? And we extended it across two little metal supports here that were capable of registering shortening or tension within the muscle. So this was an apparatus, okay? And associated with this apparatus was some type of electrode that was capable of generating an electrical stimulus that brought about muscle contraction, okay? So when you delivered that electrical stimulus, the muscle would contract and shorten. And you could record the changes within the muscle in response to the electrical stimulus. And we would call this a myogram. Okay, so the myogram reflected the generation of muscle tension versus time in relation to the stimulus, which was usually applied at time 0. So what you would notice is that within a very short period of time following the onset of the stimulus, the muscle would begin to contract. But there was a latent period between the onset of the stimulus and muscle contraction where nothing initially happened. Okay? Then following the latent period, you had this period of contraction where there was a buildup in tension until tension maximized and then the muscle began to relax. You entered into the period of relaxation. Okay? So why the latent period? Well, the latent period only exists for milliseconds, but it consists of that interval during which excitation, contraction, coupling is happening, in which the electrical signal is traveling along the sarcolemma, then down the T-tubule, and bring about release of calcium. It includes that interval where calcium is binding to troponin and causing displacement of tropomyosin. Okay? Then finally, when cross-bridge formation occurs, we begin that period of contraction, okay, where cross bridges are actively formed and we have the power stroke being generated. Okay? Now, as calcium then gets reabsorbed back into the sarcoplasmic reticulum, okay, and the binding sites are recovered, then relaxation begins. Yes?
Is this the same instance when, like, you're hurt and they put that little patch on you that, like, shocks you? Is that the same thing? Have you ever heard of that?
Like a tense unit? Like, yeah.
Like a what?
Like a tense unit?
It's not a TENS unit, no.
It's literally like
No.
a patch, and it has, like, I know because my
Muscle
friend,
contraction?
yeah, and my friend hurt her shoulder, and she, like, went to the doctor, and they were like, the only way you're going to be able to, like, heal is if you, like, stop using your shoulder, but it's the hand that she writes with and everything. So she was like, well, I can't really do that. So she, like, puts it in a thing when she can, but other than that, she, like, they told her to, like, buy the thing, and it, like literally shocks her arm.
Well, you know, it sounds like it's being used, an electrical shock there is being used therapeutically to cause muscle contraction.
Yeah.
So, I mean, the principle of muscle contraction being generated by an electrical impulse is the same, but this is being done for experimental purposes.
And that's being done.
Yeah,
for
that's
I was
being
just
done
wondering if
for
it was the
purposes.
same thing or not.
We should do that experiment.
Do you want me to steal her thing?
Now,
No.
here's the thing now. Muscles don't work the same as muscle twitches. You know, muscle twitches are all or none phenomenon. If you generate enough of a stimulus, electrical stimulus, the muscle contracts. On the other hand, in real life, muscle responses are more graded. In other words, if I were to go to pick up that marker and put it in my pocket, I wouldn't necessarily contract with full strength, bring it to my chest, and then drop it into my pocket. Okay? It would be a more graded response where I'd use just enough power to pick up the marker and smoothly bring it to my chest pocket and drop it in. So how do you vary your muscle responses? How do you grade your muscle responses? Well, there are two ways in which we can change muscle tension, that we can bring about muscle contraction of different degrees or intensity. One is by changing the frequency of the stimulation. Remember that a nerve is carrying multiple axons, each of which is branching to provide innervation to the various fibers. Okay? So if that nerve is conducting an impulse along its axons, it's going to bring about muscle contraction. If those impulses travel with a greater frequency, if there are a greater number of impulses per second, it's going to bring about a stronger response. So that's one way in which we grade our muscle responses, by changing the frequency of stimulation. In other words, with a higher frequency of stimulus, of action potentials, we release more acetylcholine that binds to more receptors that open up more chemically gated channels, okay, and do so in a repeated fashion so that muscle contraction increases. That's one way. Second is by changing the strength of the stimulus. And by the strength of the stimulus, I mean recruiting additional muscle units. The more muscle units that are involved in a contractile movement, the stronger the response. So you can either change the frequency of stimulation or change the strength of the stimulus. How does changing the frequency intensify muscle contraction? A single stimulus resulting in a muscle twitch looks like this. Okay? If instead of simply delivering a single action potential, we increase the frequency of stimuli, for instance here, 3 action potentials in rapid succession, the muscle begins to contract, and before it completely relaxes, another action potential comes along, and then the muscle contracts with a little bit more force. And before it completely relaxes, yet another action potential comes along, and with that one, still greater force. Okay? So, frequently delivered stimuli increase contractile force. This is called wave summation. Okay? Now, part of the reason for this is that in this case where we have a single twitch, the muscle, as the muscle relaxes, all of the calcium undergoes reuptake back into the sarcoplasmic reticulum. In this case, before the muscle completely relaxes and all of that calcium gets restored back into the sarcoplasmic reticulum, another action potential comes along. So there's still some calcium in the sarcoplasm when another action potential comes along and releases still more calcium. So with each new action potential, more calcium is being released, which binds to more troponin, which opens up more binding sites for cross-bridge formation. And so we have this wave summation where the muscle contraction gets stronger. Okay? And as we increase the frequency of stimuli, we develop incomplete tetanus, and we can increase the frequency until tetanus, sustained contraction, occurs. So by delivering more frequent stimuli, muscle contraction increases. Now, the second mechanism by which contractile strength increases is by recruiting more motor units. You know, with recruitment of a single motor unit, only so many fibers are going to contract. If we recruit an additional motor unit, you have an increase in number of contracting fibers, and still another motor unit, more fibers are going to contract, resulting in a greater force of contraction. So those are the two mechanisms by which we increase force of contraction. As I mentioned, if you deliver action potentials at a high enough frequency, you're going to have sustained contraction, what we call tetanus. Another important term is tone. Okay? Tone is partial contraction. sustained partial contraction of muscles brought about by spinal cord reflexes, okay? Such that one group of motor fibers or motor units are activated, okay? Others are not. As that one group of muscle fibers contracts, it triggers spinal reflexes that bring about then relaxation of that first motor, those first motor units, and then contraction of a second group of motor units. And so that there's an alternation between one group and another group, so that you have partial sustained muscle contraction that brings about a certain amount of tone. As you're sitting here, okay, sitting down at your desk, you're exhibiting partial muscle contraction in your paraspinous muscles. That means that alternately you're sending signals to some motor units to contract, and then those remain contracted for a small period of time, and then they elicit another group of reflexes that bring about relaxation and then contraction of another group. So at any one time, there are a small group of motor units that are triggering contraction, whereas others are relaxing, and it's alternating throughout the muscle. This tone keeps the muscles firm so that it can respond to stimulation. Now, if you have injury to a nerve, you lose muscle tone. Okay? How can you tell if someone loses muscle tone? If you lose all innervation to a muscle, and I were to pick it up above your head and drop this hand, what would it do?
Just fall.
It would drop and hit your head. As long as you have partial tone, if I let go of this hand, it's not going to hit your head. You have enough muscle contraction that you'll avoid your head.
It would like death fall on your head, like you wouldn't like fall
Yeah, if
backwards.
you have someone who
or-
has a completely denervated upper extremity, let's say they had some type of plexus injury, okay? You lie them in bed and you put the hand in front of their face and drop it, it's going to hit the face. There's absolutely no tone, no muscle contraction. Whereas if there is some partial contraction or tone, you let go, then more than likely they'll kind of just move it away from their body so it doesn't strike their face.
Okay.
So muscle tone refers to partial contraction. Okay? That's brought about by spinal cord reflexes.